Dr. Zelko Bosjnak, who's connecting from the Medical College of Wisconsin at Milwaukee. Again, I want to say it's around bedtime for him, but I've I've forced him to sit without sleeping, but I'm sure he's going to be awake once he starts talking. And today is the first lecture today in a day two. So this the research, I'd like to first introduce him to all of you. He's one of the finest researchers that I know. He's a universal researcher. He has collaborations with almost all the countries, I would say, with Japan, and he travels to other Croatia and other places. So his main research areas are mechanisms of anesthetic cardioprotection, biomarkers of anesthetic-induced neurotoxicity in children, surrogate biomarkers of adriamycin-induced cardiotoxicity. So these are the major areas that he is currently focusing. So he received his doctorate degree from the Medical College of Wisconsin, and he's been with the Medical College for about 40 years now, which is a very long time, long association. So each one of us who were working there knew about him, knew him as a very cordial person, as well as he was kind of the superstar of Medical College of Wisconsin, because he was the one person who had great grants all the time throughout the period when a lot of people weren't able to get grants. He was the one who used to get a lot of project grants, and he has more than 320 research articles, reviews, books, and he has a lot of credentials. The best part that I know him of is he has trained more than 100 postdoctoral students, which is a phenomenal achievement, I would say. And he has served in various committees of MCW. He is the editor of various excellent journals, including the anesthesiology and stuff like that. So he's a long-time member, and he's also the chairman of the NIH Surgery Anesthesia. And to be a chairman of NIH, it needs a lot of credentials and knowledge, and he's an important part of the study section of NIH. And he's a member of the National Advisory General Medical Sciences Council, and he continues to serve in many sections for the NIH. He has various projects, and his lab is constantly busy churning out a lot of very good papers. And it is really my honor to having been collaborating with him. I had a fantastic student, Tarun Pant, who went on to get trained with him, and he's currently a postdoc with Dr. Bosjnak, and they are doing very good work. And I get opportunity to visit them quite often and get a slice of what they're doing. Dr. Bosjnak has received several awards for his research, including NIH New Investigator Research Award, Research Career Development Award, and American Society of Anesthesiologists Excellence Award, and so many other awards, including the 2 presidential medals from the National Decoration for Science and National Decoration for Humanitarian Work. So this is what makes him a great person, not only being a great scientist, but also being a very passionate researcher and a great humanitarian. So we welcome Dr. Bosjnak today, and I'm really, really, really grateful to him for having accepted this offer to meet all of us here and giving his inputs on what kind of research he's been doing. And thank you so much for obliging, as always you do for me. Thank you, Bosjnak.
Thank you very much for such a kind introduction. Indeed, good morning, everyone. I'm delighted to participate in this international webinar organized by Biomedical Technology for the Future here at Center for Biotechnology at Anna University. So I will go ahead and try to share my screen with you. Oops. Host disabled participant screen
I can.
sharing.
1 second, just give me 1 second,
Okay.
sorry.
All right.
Yeah.
Am I okay?
Yes.
All right. Very good. All I have to do is just find my stuff now and share. Are you able to see?
Yes, yes, I'm able to see, but
Okay.
can you
Very
make
good.
it? Yes, perfect.
That's about as large as I could make it on my screen. So this morning,
Yes, yeah, perfect. Yeah, it's perfect.
very good, thank you. So this morning, I'd like to share with you one part of our interest, and this has to do with pediatric anesthesia that might potentially cause developmental neurotoxicity in young children. So what we are essentially talking about is less than 3 years of age of a human brain during the so-called brain burst and intensive synaptogenesis, essentially. So what I'm going to share with you, not only our research, but research from other individuals to point whether or not indeed these very powerful agents, when given during this such a young age, might adversely affect the development of a human brain. Now, as you know, a lot of children at this age, young age, during their brain burst, do require surgeries. Either that or life can be threatened, obviously. So surgeries are necessary. But what I'm going to show you today is the fact that some of these powerful agents might adversely affect the development of a human brain, one. And two, even more important, are there other anesthetic agents that we might be able to use in the future? So to conduct surgeries, but without some of these developmental neurotoxicity side effects themselves. So several years ago, the Food and Drug Administration issued a warning here in the United States in December 2016. And what this warning said that repeated or lengthy use of general anesthetic might cause in these younger children, less than 3 years old, to have some developmental problems later in life. Now, the FDA raised these concerns, but did not have many answers themselves. They went further to say, to give a suggestion to parents or caregivers that they should discuss this child's health care with professional about these potential adverse effects. And also for pregnant women, because some women, as you know, need to have a surgery during various trimesters of their pregnancy, and a similar conversation should be held with pregnant women as well. So FDA issued this huge warning, but to be very honest with you, we at that time did not have enough data to really justify this warning. So there was a lot of panic, obviously, in the clinical community and when the children need to be anesthetized, because parents obviously worried a lot whether or not they should even have tonsillectomy on their children and so forth. So this morning, I'd like to list several learning objectives that we're going to share this morning. One, we're going to talk briefly about some of the preclinical findings of anesthetic neurotoxicity. In other words, some of the animal work. We're going to do that very, very briefly. This would be followed by some of our input on mechanism of anesthetic toxicity using a stem cell biology. Then we'll follow into the clinical studies and talk about retrospective and prospective epidemiological clinical studies. In other words, what is being done out there, okay, to address this problem. And then I'm going to close on future clinical trials with potentially different anesthetics or so-called apoptosis sparing anesthetics that might help us to prevent this damage of a growing human brain. Now, during our development, it turns out that most of our neurons or progenitors, with some estimates up to 70% of those neurons and progenitors, are actually programmed to die if developmental milestones are not achieved. That means that most of those neurons are going to die in a normal course of the development. And as a matter of fact, if one were to prevent that programmed death of the neurons, one has totally abnormal brain. What's the problem with anesthetics? Well, it turns out that anesthetics affect some of these milestones during the periods of brain burst or synaptogenesis, very intensive synaptogenesis. Alcohol does the same thing, anesthetics do the same thing, and we know what alcohol could do in a fetal alcohol syndrome. It turns out that these powerful agents, including alcohol, act on mostly two channels and receptors, NMDA and GABA-A receptors, okay? So NMDA antagonists and GABA agonists, that's how actually anesthetics work. And so that is the problem that I'm going to show you as we go now further. So anesthetics, and I hope you can see my little pointer here. On the left side, I have an examples of some of the general anesthetics. Most of them are inhalational, but we do have barbiturates, etomidate, and propofol. They act on GABA-A, and they inhibit, they excite this inhibitory channel itself, where chloride goes, once it's open, chloride fluxes inside the neuron, hyperpolarizes the cell, and inhibits the activity of the neuron. On the other hand, some of the anesthetics, ketamine, nitrous oxide, xenon, and so forth, they act on NMDA receptors. These are excitatory receptors, having calcium flow into the cell, and they would basically inhibit this. Some of the inhalational anesthetics actually affect both of these channels themselves, okay? So that's how most of those anesthetics end up working, without just about any examples. What is the difference between immature and mature neurons? Especially this one here on your left, we have immature human neurons. So these are not just a small little baby neurons, they are immature. They are still in the process of development, process of acquiring proper channels, proper exchanges, and things of that nature, and proper concentrations of ions themselves. So here on your left, we have a GABA-A responsive neurons. And it turns out during the development, during the synaptogenesis, intracellular chloride concentration is relatively high as compared to your right in a mature neuron where chloride concentration is relatively low. So what happens if one activates GABA A receptors in an immature neuron? It turns out that chloride goes out of the cell, all right, effectively causing greater excitation of the neuron. In the adult neuron, and I'm just showing you one example, in the adult neuron, when this channel opens, chloride fluxes down its concentration gradient, if you will, okay? So it's not a total membrane potential, but it's a concentration gradient that depends on the concentration and membrane potential and equilibrium potential of those ions themselves. So in a mature neurons on your right, activation or opening of this channel will cause hyperpolarization and inhibition of that particular neuron. While on your left, in immature neurons, this flux of chloride will be going from inside out, causing depolarization and excitation of the neurons. So without making this too complicated, the fact of the matter is that excitation of the neurons also part of this apoptotic nature and triggering for these neurons to ultimately undergo a programmed death. So it is likely that this excitation of the immature neurons during the synaptogenesis is the reason for triggering this apoptosis. So what we're trying to say is that these very powerful anesthetics are going to have differential effect on these neurons, depending upon whether or not they are immature, undergoing synaptogenesis, or if they are mature. It turns out that once the brain matures and the child is older than 4 years old, these anesthetics are going to be hitting more mature neurons, and there are no detrimental effects of general anesthetics. So these studies have started a number of decades ago, even 1981, where these are first reports that these inhalational anesthetics, halothane and fluorine, affect the learning function in mice, okay? And then but nobody paid any attention to this. 10 years later, they showed the same thing in rats. Well, nobody paid much attention to that either. But interestingly enough, in 2003, a study by Yevgeny Tudorovich in 2003 showed exposure to the common anesthetic agents in rats causing, again, problem in developing of these animals. And it is this last study that sort of kind of put the whole concern on a front burner. And a lot of studies were done after this publication in 2003. And rats, of course, mice, other rodents have a different brain structure, development that's much more compressed. And then it is very important that subsequently in 2007, 2010, 11, and so forth, studies on non-human primates actually were done as well. Here, showing that ketamine induces cell death, isoflurane neuroapoptosis, ketamine, and so forth, using rhesus monkeys themselves, okay? So now that is those studies are extremely important. Why? Well, because this brain growth, brain spurt, synaptogenesis in humans mimics human brain development much closer as compared to the rodent's brain. But even so, there are certain differences in human versus, for instance, a monkey brain, whereby here's the birth indicated here with a vertical line here. And you can see that monkey brain at the time of monkey's birth is quite much more developed, if you will, as compared to the human brain. Human brain, actually, after the birth, has by far much longer to go in terms of the development itself. And by that alone, it is going to be much more sensitive to these powerful agents when given here sometime after the birth and prior to, let's just say, 3 years of age. So here's a fetal apoptosis pattern after 5 hours of isoflurane anesthetic in monkeys. And here's on the left is a control apoptosis. So this is a normal apoptosis of neurons and oligodendrocytes, red neurons, white oligodendrocytes. Oligodendrocytes, as you know, being responsible for myelination of exons. After 5 hours of isoflurane, the pattern of apoptosis is similar to control. However, you can just see it is hundreds of times much more intense as compared to control. So that's a slice of the brain here, and cerebellum and the base brain stem. You can also appreciate how great of the apoptosis this is. And sure enough, in these monkeys, there is a neurocognitive dysfunction as it goes. So without belaboring the point, the conclusions from all of these animal data, and there are many of them, that show that in developing animal, be it mice, rats, monkeys, and so forth, when they are given anesthetic, they will have early activation of neuroapoptosis. Many neurons are going to die, leading to later onset of memory and learning abnormalities themselves. Okay, so those are the animal studies. We came into this field by actually using patient-specific stem cells. We used induced pluripotent stem cells because of some of the ethical concerns of using embryonic stem cells. We use induced pluripotent stem cells from the patients. This is where, as you know, patient cells, skin fibroblast, blood cell, even cells that are in urine of the patient could be reprogrammed with these factors to become induced pluripotent stem cells from any individual for that reason. And once we have these stem cells, they can, in the right condition, they can expand and divide indefinitely, making absolute copy of themselves. And once we have those stem cells with appropriate growth agents and stimuli, we could basically push these cells into various differentiation, be that liver cells, nerve cells, cardiac cells. Most of our research, as indicated, deals with differentiation into cardiac cells, so we can use these patient cells to do a variety of things, from toxicity studies, precision medicine, and so forth. But for this particular study, we used nerve cells, basically. We used the protocol to derive a pure dish and differentiation of the nerve cells. It takes about 36 days, whereby the human stem cells undergo different differentiation from embryonic embryoid bodies to rosette cells, neural stem cells. These are progenitors that are going to ultimately be specific into the brain cells themselves, and then finally into the neurons, astrocytes, oligodendrocytes, work done by Dr. Bai and others in our laboratory. Now, in order to be sure that we are getting the right population of cells, we have to obviously not only label and be sure that we have a proper induced pluripotent stem cells and that they could indeed form teratomas. So we would have to carefully label these cells to make sure that these are iPSCs. Then we go into the rosette cells themselves with the various markers, and then go further down to neural stem cells themselves with various immunohistochemistry labelings, and finally get down to the neuron themselves, whereby we could be sure that our culture is indeed pure human neurons themselves. And then they are immature neurons, and they are perfect for us to actually study how anesthetics are going to affect immature neurons. This is the time when they are forming synapses between each other. Double cortin staining indicates migration or an immaturity of these neurons themselves, and so that we can then go ahead and test anesthetics during this critical phase of brain development. Moreover, we have to be sure to label and get the handle on NMDA receptors, and here's the staining for that. What about GABA A receptors? They have it. So without these, obviously, two receptors and channels, we would not be able to test the potential effect of general anesthetics. And that, of course, was also documented to make sure that we can actually even record NMDA and GABA A currents from these neurons. So then, once we have these pure cultures of neurons, monolayers of neurons themselves, then we can go ahead and test the clinically relevant doses of anesthetics and look at the different exposure times. We can do short exposure, let's say less than an hour, and that's what most of the clinical studies have done, less than an hour, unfortunately. Or we can look at different frequencies. We can expose them to short duration, but several times, or longer duration. And then look at the cell viability, not only in terms of tunnel staining and the death of some of these apoptosis of these neurons, but also we can look at the structural components themselves, because it's not only the death of the neurons, but clearly these anesthetics affect the development of these neurons as well. So over the years, we looked at the many different aspects of it, including mitochondrial dynamics as well. And I will try to play this video here. You could actually see that mitochondria, even in neurons themselves, are very active, just like they are very active in a mitochondrium of the myocytes. They are labeled here with the green fluorescence. Using computer software, we highlighted artificially a couple of these. Here's the yellow one, here's the red one, and they will end up merging and things of that nature. To make a long story short, we were very much interested into this balanced cycle of mitochondria. They undergo fission fusion type of a cycles, and this is a normal way the mitochondrial dynamics occur, not only in human neurons but also in other cells. And it turns out that following the anesthesia, mitochondria undergo much greater fission status as compared to uh, um, fusion. So we saw an increase in mitochondrial fission, and here's a control labeled, green fluorescent labeling on your left, control, and we have electron microscopy as well. Following exposure to one IV anesthetic propofol, you can see that fragmentation of the mitochondria in both after green fluorescence as well as EM. If we prevent fission with mitochondrial fission blocker, MDV1, we see that it turns out apoptosis is not going to occur, and mitochondria will be more normal, if you will. And why is this happening? Well, it turns out that fission itself actually ends up hyperpolarizing the mitochondria, causing greater amount of reactive oxygen species to form in the presence of anesthetic. And then we went on and we looked at that fission on your right here, as well as a role of various microRNAs, and we assigned specific PDCD4 as well, and then of course fission, as I just mentioned. This is where it goes from the fusion to fission, and the greatest amount of fission ultimately ends up causing this reactive oxygen species. So those are the mechanisms that we have studied, but that is still not going to be enough for us to say with a certainty whether or not these changes are actually responsible for developmental problems and cognitive impairment of the brain itself. Nevertheless, there's one very critical component that came from many studies that we published, and it's summarized really in this particular slide. So here are the agents, both intravenous as well as inhalational anesthetics on your left: propofol, ketamine, isoflurane, sevoflurane, desflurane. Any of these agents that act on either or both NMDA or GABA-A are going to cause neuronal apoptosis and death of the neurons. Agents here in the middle, like dexmedetomidine, this is an alpha-2 agonist itself, also causing anesthesia and analgesia, or remifentanil and other opioids. They are affecting the brain not by NMDA, GABA-A mechanisms, but by other various other receptors and mechanisms. Interestingly enough, they do not cause problems during the brain development, okay? Of course, time is involved as well. Shorter duration that we have shown, 1 hour, are fine, but anything over or multiple exposure cause the difference. So it's not just an agent, it's also duration itself. Nevertheless, these compounds and others do not cause apoptosis. Then we went a little step further, and instead of just having a single layer cells, we generated three-dimensional model of human brain itself. Okay, so we essentially, like a form a mini brain. It's about 3-4 millimeters in diameter, and here's first published in 2014. So soon after that, we tried as well, and we did it through bioreactor. It does limit its size of the growth of this brain. But the nice thing about this is that it contains all of the other cells that are going to be a part of the brain. So it kind of models the human early development itself and recapitulates various brain regions from forebrain, choroid plexus, hippocampus, and so forth. And with various labeling, one can clearly see various progenitors, neurons themselves, and they can mimic in vivo organ physiology. So it's not just a single cell, but now we're talking about various other cells that are part of the brain itself, which might actually indicate differential response to the agent themselves. And so if you look at, here's the mouse brain development versus the human brain here on the right. So when we talked about these progenitor cells, here I indicated in this, in the lower part, and this is our progenitors that are going to basically make a daughter neurons themselves. And these neurons will climb up the apical radial glial cells themselves and form the various layers, okay? So these are the oldest ones here that indicated in dark, and the neurons here are indicated in a lighter shades themselves. So these new neurons actually have to work themselves through this little forest and form a new layer. And one can do a lot of different studies using this type of a model. Here, I'm just kind of a schematic component showing you the same thing. If one labels these progenitors here with BRDU, they will end up making hundreds, if not thousands, of daughter cells that are going to migrate now. This is a mouse brain, migrate, and if you label it, here they are, in this particular case, they are migrating to a certain layer. Subsequent cells, without labeling, will go ahead and pass and go past this and be up here. Now, this is where one could look at the anesthetic. In this particular study, they have actually looked at the ultrasound. How does ultrasound during the pregnancy end up altering the brain itself? Well, during the ultrasound, you can see that some of these neurons are going to actually not migrate at all. And some of those that are migrating will be actually perhaps in a different locations themselves. So this three-dimensional model gives us additional tools, if you will, where we can actually study the actual human composition of the brain. So clinical relevance is very high, and this is where one can look at the various drugs during the development itself. And just for comparison, here's the mouse brain on the left. Red stain are progenitors. Green are the neurons themselves after migration, if you will. And here's a 3D organoid from the laboratory. Here are the progenitors and migrating neurons here forming the layer. So there's a lot of things that we can actually do with this model. And one of our graduate student, postdoc, Sarah Logan is very much interested in this. Okay, so that's about preclinical as we can go. What about clinical studies? Now, studies on population-based studies are mostly retrospective. So, and they don't have much in terms of numbers and so forth. Some of them are showing increased risks, some no increased risk uh, uh, when they studied retrospectively. Nevertheless, increased risk, they have shown more multiple exposures to anesthetics, okay? In other words, they studied more multiple exposures. Most of, all of these actually studies have actually only used short duration of anesthesia, short anesthesia. We in the lab have also shown that the short duration does not cause much of a problem. So right studies really have not been done. Nevertheless, multiple exposure of anesthetics have been documented from Mayo Clinic that there are learning disabilities if a child receives more than one anesthetic exposure. Now, lately, we have seen more current large clinical reports that are prospective, okay? So some of these are good. The problem is it was a short anesthesia again, all right? So, and they have not really shown a heck of a lot difference in terms of cognitive function of the children 2, 4, 5 years afterwards. Nevertheless, this last study that has quite a few kids in this particular study has shown attention deficit hyperactivity disorder appears to be much higher in exposed children as compared to those that are unexposed. Okay, so there is a considerable hint that there is a problem even with short duration anesthesia. Now, I'll show you one slide with 1.5 million anesthesia duration, just so that we can get some idea what are the duration of anesthetics in children. Most of the 94% are below 3 hours, as you can see. These are vertical lines. This is 1 hour, and this is 3 hours. So most of the studies were being done with these short duration, less than an hour exposure. But clearly, there is a huge number of children that have to undergo, because of the, let's say, cardiac surgery, reconstructive urinary bladders, things of that nature, surgeries, orthopedic surgery, that are much, much longer. And I think this is obviously the population at risk, if you will. So one of the things that we've done is to look at some of the exosomes that come from the brain and into the blood effectively. So we looked at children who had a short duration anesthesia and long duration anesthesia. And we wanted to see if we can actually detect some of the biomarkers in their blood, okay? And what we did is we listed a whole bunch of biomarkers of our interest. Some are going to be helpful. Some will indicate that there is a potential damage to the human brain. So what we did, we took a blood sample before the surgery and blood sample after the surgery. And Dr. Tarun Pand, our postdoctoral fellow here in the lab, is leading this particular study. It's not done yet, but the idea was to actually look at the kids who are less than 4 years old and having surgeries less than an hour and greater than an hour, 2.75 to 3 hours surgery. And then look at the kids who have already mature brain, up to 17 years old, okay? And so the idea is, our hypothesis was that kids who are less than 4 years old, we will see the differences between short duration surgery and longer duration surgery. And so far we have, and then of course we have also cardiac and non-cardiac surgeries. So far we have limited results because they are ongoing right now, and because of the hibernation, because of COVID, everything is kind of put on a back burner. Here's a relative gene expression that Dr. Pant did, mRNA expression. And what we have seen, here's a group A, green, constitutes a short duration surgery in a young population, less than 4 years old. And red bars is group 2, that is a longer duration surgery. Green is short, red is long. And what we saw here for the mRNA, we are doing the proteins are going to be done hopefully sooner rather than later. We see that myelin basic protein showing myelin breakdown is higher. Neuron serum amylase, indicator of neural injury, is higher. Here's a calcium binding protein B, indicating acute phase of brain potential damage, is higher too. So we are very anxious to see what actually happens in the older population. We obviously hypothesize that that is not going to be as drastic as this particular. And I think these biomarkers are fine, okay? And a lot of good information will come. But the question is, where do we go from here? And this is where I'm going to, in the last 5 minutes or so, give you an example where we need to go in order to ultimately solve this particular problem. In other words, we need to establish this definitive link between anesthesia and adverse neurodevelopmental outcomes. Now, studying the very same anesthetics that are causing the problem, it's very hard to prove this negative, if you will, okay? And so we need to basically use not only just a standard, this is on the number 1, standard anesthetic technique, but we also have to change anesthetic strategy and use agents, anesthetic agents or sedatives, that are not going to affect NMDA and GABA A mode of action. That's the key, all right? So we need to have randomized clinical trials and then basically compare the children that are anesthetized with standard technique and compare these children having the same or similar surgeries using completely different anesthetic, getting away from NMDA and GABA A. And this is where my colleague, Dean Andropoulos from Houston, has actually tested these different anesthetics, including a local so-called T-Rex study, where they have used alpha-2 agonists, dexmedetomidine, they use opioid, remifentanil, and when necessary, use also caudal anesthesia with bupivacaine. So these are the sodium channel blockers. So staying away completely from NMDA, GABA A. And that's the only way to ultimately test under longer anesthesia whether or not there are differences between these two. And what's very important is that most of these infants could tolerate these anesthesia regimen, okay? Sometimes it is necessary to very little bit supplemented with inhalational anesthetics that are not going to be that are not going to be toxic. So currently, that's what's going on. As a matter of fact, they are now testing standard anesthesia with this apoptosis sparing agent. And that's the only randomized clinical trials that will ultimately answer that particular question. So I would like to actually summarize very briefly what are some of the things that we talked about. Preclinical studies are very firm on their outcomes. There's no question. They are very solid and very well documented that in animals, during the brain burst and synaptogenesis, it will cause detrimental effects. Epidemiologic studies themselves are a problem because they're all short, and some that show problems, some don't show problems. So, but nevertheless, based upon this, FDA came through and gave us a warning 4 years ago. We are looking at the biomarkers and exosome. That's all fine. It's going to be nice chain, ultimately leading to these randomized clinical trials using 2 different anesthetics. And basically that is ultimately going to be able to tell us whether or not there's a problem with NMDA GABA A type of anesthesia. So in a summary, preclinical findings, there's a strong evidence themselves, especially with the multiple procedure and longer procedures. Retrospective clinical findings are conflicting. Nevertheless, with multiple exposure, there is a problem. And these prospective epidemiological studies showing, very good studies, showing that 1 hour exposure is fine. But it is these future clinical trials, I believe, with apoptosis sparing agent that are ultimately going to help us answer that question if the anesthetics are toxic to the growing human brain. I'd like to thank you for your attention, and if there are some questions, I would be happy to attempt to answer them. Let's see.
Thank you very much, Dr. Bosjnak, for the insightful talk. It has always been a pleasure to listen to you, and the way you put things very clearly and that everyone can understand who have not even been in the field. I'm sure everyone would have understood what you've been trying to say. So let me first ask the questions that people have put forth here. And so what do you think could be a possible alternative for these general anesthetics, sir?
Right. So, as I mentioned, there are several candidates. Unfortunately, we don't have a good general anesthetic that is not going to be acting via NMDA and GABA A. So they usually have to be supplemented. So you take your alpha-2 agonist, okay? By itself, it's not like a propofol that would anesthetize people. But that, along with opioids, is pretty good anesthetic as a matter of combination of anesthetics, apoptosis sparing in young children, okay? So chances are it will be a combination of alpha-2 and opioid anesthesia, even if one could use maybe local anesthesia and caudal, but that's what's going to be. Now, I happen to know that there are several laboratories who are developing a brand new anesthetic, not the, from the toolbox that we have, but they are developing some specific neuronal calcium channel inhibitors that might prove to be rather good in the future. But as of right now, today, what can be used, it's probably a combination of opioid and alpha-2 agonists like a dexmedetomidine.
Okay, thank you. And the next question, what techniques are you using to study the biomarkers? Is it protein microarrays or some other techniques? And how are the biomarkers collected?
Right, right. So Dr. Pond has just looked at messenger RNAs right now. With ELISA, various ELISA assays, our next step is going to be to actually look at the proteins themselves that are likely to be in that cargo in exosomes themselves. So mRNAs and ELISA assays for the protein screening. Later on, we might use some fancier techniques, but ELISA and mRNA techniques right now for general screening between young population and older population.
Okay, the next question is there any difference in the effect of local anesthetic versus the general anesthetic on the brain?
Right, very good question. It depends on the concentration of local anesthetics. Generally speaking, local anesthetics, because of the way they act on specific channels of axonal transmission, they pretty much do not affect the period of synaptogenesis and growth itself. So they don't appear to be detrimental to the newly forming brain itself, unless the concentrations are huge and so forth. They too might be detrimental via different mechanisms themselves. But they are clearly safer if used at the clinically relevant concentrations as compared to general anesthetics that act on NMDA, GABA-A themselves, especially at the longer duration.
Thank you, Dr. Bosjnak. I have a couple of questions. One is, you're talking about these apoptosis sparing agents. Have any other supplementations been tried, or would those work? Is there any studies on those areas, like supplementing this with some other, agents?
Well, sevoflurane is used a lot, obviously, in children. It has to do with better acceptance as compared to isoflurane and halide and so forth. A number of studies have actually shown that supplementing these general anesthetics with dexmedetomidine, alpha-2 now agonist, supplementing appears to attenuate the toxicity of these anesthetics, mostly in preclinical studies showing that developmental impairment appears to be decreased if these anesthetics are supplemented with dexmedetomidine. What Dr. Dinandropoulos is doing is obviously not even supplementing, but using dexmedetomidine itself, which seems to be counteracting sevoflurane effects, to use that with opioid instead. So there are some attempts being made to supplement these general anesthetics with agents that might mitigate potential toxicity itself.
Okay, and I have one more question. So you're saying that these infants are getting ADHD and those kind of issues. So is there any comparative study between the infants who get ADHD through this neurotoxicity versus, ADHD due to other reasons? Is there any gene expression studies or biochemical or neural activity network studies which have done between these two groups?
Yeah, it's a very good point. Unfortunately, very little has been done, especially comparing younger versus older children during these anesthetic periods, especially uh, uh, during these longer anesthesias. We feel that short, less than an hour anesthesia are really no problem. But the longer the anesthesia, the bigger the potential problem. We are trying to, as you can tell, looking at some of these biomarkers that end up crossing blood-brain barrier via exosomes. So exosomes can cross the blood-brain barrier without much of the problem. Actually, it happens all the time. But even our study is not going to be definitive. Definitive is going to be direct comparison between apoptosis-sparing agents and current clinical practice with general anesthetics, which is very easy and easier than these apoptosis-sparing agents. So we have obviously ways to go before we ultimately answer that very critical, clinically important question, whether or not they are detrimental during the long anesthesia.
Thank you. And is there any report about the mortality rate? So you keep talking about the neurotoxicity, but what about the mortality rate because of this anesthesia? Is there any chances of it being causing mortality?
Yeah. Well, the mortality is not as big of an issue as is cognitive impairment, itself, because the sheer damage to the neurons and plasticity of the brain, brain can take quite a hit, if you will, and recover. But that recovery period is going to be very much also dependent on enriched environment. And that's been shown in, animal studies. After the same anesthesia, if you put animal, in this particular case, into non-enriched environment, just the four white walls, they will have a greater problem because of the anesthesia as compared to another animal that is in enriched environment. But mortality, no, at least not during or shortly after the surgery. Usually the surgical problems are the ones to end up causing the fatalities.
Thank you, Bosjnak. And I'm sorry, one last, I have a lot of questions, so
Sure,
one.
sure.
So this study on organoids is very interesting. I mean, it's a known fact that it kind of mimics the human brain, mini brain. They are like, even in their culture, they look like mini brains. In fact, this was one of the questions by the participants, if they can relate. Yes, I mean, people have been using organoids as a great model. There's no doubt about it. But yeah, but I've read that it takes at least 2 months to even see small oscillations and the rhythm spikes and the action potential and stuff like that. And to look at actually the neural network activity, it takes about 3 months. So I think it's quite a lot of time to do that. And do you think, is there any other alternate methodology that can be used to kind of mimic this situation rather than waiting for 3 months? And if something goes wrong, you have to start from scratch all over again.
That's right. So it's a very good question, and you're right. There are clearly limitations of one using this type of thing. The idea is to continuously, I guess, keep on producing these organoids. I think that the slicing and doing the brain slices is one of the ways to get closer to the intricacy of disorganization following so yes, it is complicated. It is complicated, and there's just so much information that you can extract using this particular technique. in some of the ways, in some of the methodology, it's easier than others, but it's clearly not an answer to everything. And I don't know of any other model at this particular time to help us out.
Okay. Okay, thank you. One last question from the audience. Are there any solutions to come across the BBB problem using stem cell techniques? I don't understand.
Okay. Is there a solution to come? I'm not quite sure what the question is.
What is this BBB problem? Blood-brain barrier, you mean? Maybe?
Yeah, I think so. So one of my previous graduate students went to Madison and worked with Sean Polachek in Madison, Wisconsin. They used the stem cells. They used the stem cells to actually build a blood-brain barrier, and so it's still a very beautiful way of testing various drugs, permutations, and so forth on effects on blood-brain barrier, especially when it comes to cancer drugs and things of that nature. You know, the permeability of various drugs is extremely important. And so having a human-derived BBB is very important. And one can, of course, test the permeability by actually measuring the substances before and after, but also the impedance, in other words, impedance between high impedance that these stem cell generated BBB can produce. Nevertheless, as I said, the transport of the signal molecules via exosomes is pretty much straightforward. It's just about diffusion, if you will, across, and that's why we are using these biomarkers even with intact blood-brain barrier itself.
Okay, and I have one more question. Does this neurotoxicity cause any retinal cell damage?
This is a very, very important and question indeed. And as a matter of fact, a number of investigators have looked at the development of some of the neuronal endings in the eye during the development and how anesthetics actually can affect. So anesthesia can definitely affect cells in retina as well, but it depends at what point in development they are. You saw in a brain those sections that I showed you, sections of the brain, and how the apoptosis varies. It's not uniform, but it's clustered, okay, in a specific areas of the brain. And why is that? Well, during the anesthesia administration, those areas appear to be most active in terms of synaptogenesis and so forth. The same thing is in cells of the retina as well. It depends if they have already developed, for instance, at that particular stage, if they are developed, anesthetics are not going to do much, okay, to those cells. However, if you hit with anesthetic at the time when these cells in retina are still immature, if you will, okay, chances are there's going to be greater problem. So it just depends at what point in development, if you will, are these neurons and cells themselves.
Okay, so that was a question from the PhD student who's just joined me recently.
Okay.
So she has another question. So is there any comparative studies of monitoring mitochondrial biogenesis in vitro and in vivo in anesthesia and neurotoxicity?
Oh, I'm sorry, the sound,
Is there,
I lost your last sentence, yeah.
Yeah, is there any comparative studies of monitoring mitochondrial biogenesis in vitro and in vivo in anesthesia neurotoxicity.
The biogenesis you're talking about of
Yes, yeah,
mitochondria.
biogenesis.
Uh-huh.
So is there any comparative study being done in this kind of neurotoxicity to compare between an in vivo and an in vitro situation?
Uh-huh. Very good. Not that I know of, unfortunately. Not that I know of. That doesn't mean it's not out there somewhere, but uh, the studies so far done on neurotoxicity have pretty much looked at these retrospective components that are, most of those studies are worthless. As far as the cellular effects, with the cranial windows, being done and so forth, general descriptive, but not so much at the cellular level as compared to the cardiac tissue and some other tissue. The brain, pretty much, it's much more difficult to study, and very few people actually are doing that. Our lab is probably one of the very few to actually look at some of these cellular effects and the mechanisms themselves, the pathways themselves, and so forth. And I wish we could do more, but we are now more in cardio-oncology and things of that nature.
Thank you. Lokeshwari, I think you got your answer. And the last question by Satya is, neurobasal medium is generally used for synaptogenesis in the in vitro neural cell cultures. So is it possible to somehow use this neurobasal medium to regenerate the synapse after neurodegeneration?
Well, absolutely. As you know, our brain regenerates all the time. It has a considerable amount or number of progenitors and new cells are produced. These media absolutely can be used as a neurotrophic, actually, factors themselves. Studies have shown that even using soup of the stem cells injected in certain parts of the brain end up indeed regenerating by introducing these various growth factors. So, of course, this is a way open field, but could that be done? Absolutely.
Okay. That reminds me of the stem cell therapy for Michael Schumacher. And they have said that it's working on him and he's, getting conscious.
Yeah. You talk about this
Formula One racer.
Yeah. Philippe from Alabama, Birmingham, Alabama, working on a Schumacher itself. Yeah. So it's it's a difficult case, and I think his prognosis, unfortunately, being so many years after this terrible skiing accident, I think it's extremely challenging. But that doesn't mean that different parts of the brain cannot be awoken, if you will, and retrained themselves. I just had a case where completely left side of the brain was gone, but the patient is now walking, and talking himself. So a lot of things can be done. We are at the infancy of brain generation itself as of right now.
Okay, like I was very excited when they said stem cell therapy has helped Schumacher to a great extent, and
I don't think this is happening in Schumacher's case, by the way, but I certainly pray that I'm wrong in this particular case. And I know Philippe is working very hard to help him, and I'm sure everything that could be done is being done for him.
Okay, thank you so much, Dr. Bosjnak, and I can't thank you enough for being awake so late. And I'm sure I'll be back to Milwaukee sometime when the Italian restaurant opens.
Please come. Pleasure was mine.
Thank you so much. Thank you.
Thank you
Bye-bye.
very much. So
Bye.
long.
Yeah, you too.
Bye-bye.